and I introduce the speakers, I'm just going to go over a couple things that we typically go over before we get started. I want you to, at the bottom of your screen, you'll see there's a webinar support number if you need it. But during the webinar, as long as you're logged in, you should be able to ask us a question at any time. So if you look at your navigation bar, there is a questions drop down where you can ask a question. If you have one about audio, if your screen isn't working, feel free to ask us a question there. Your audio controls are muted. We do have a lot of people on this webinar, so we can't actually have questions asked out loud over the phone, but we will do a Q&A session, which I'll get here to in a minute. The webinar slides are available for handout. They're in the handout drop down right now. So if you look in your navigation bar, if you haven't received a copy of those, we did provide those for you. And to ask a question, again, at any time, we are going to have a Q&A session from 3 to 3:30 if you'd like to stay for that. So at any point during the presentations, feel free to ask a question of either presenter, and we'll get to those questions, as many of those questions as we can during the Q&A session. Okay, and as you see here on the slide here, a training certificate is available for this webinar. If you're looking for a training certificate and did not indicate that on your registration, you can let us know. Or if there are people in the room with you who would like a certificate, you can put those names in the questions box, and we'll make sure everyone in the room gets a certificate and we typically send those out by the end of the week or early next week, and you'll receive those via email. You must attend for at least 60 minutes, and the names, like I said, the names of the people who are not logged in or in the room with you, you just put those in the questions box and we'll get the certificates to them. We do record these webinars and post them to our website, webinar website, but you have to be attending the live webinar in order to receive a training certificate. And just here, I'm not going to go over all of these, but you can see here we do have quite a few webinars coming up in late May and June that you might be interested in, including our next webinar in this small system series, which will be on June 25th on sanitary surveys. And registration will be available for that webinar here shortly. Also, just another shout out for our workshop that's coming up. It is September 24th through 26th in Cincinnati on small systems. Registration will be opening this week, so look for that. You will receive an email invite for that workshop. And we also are still accepting proposals for presentations until the end of this month. And travel may be covered for presenters, so keep that in mind and check out the website and see if that's something you're interested in. And just one other, this is just our disclaimer for our presenters. And again, before we get started, if you joined us after our start time, if you have more than one person in the room with you, if you could type that number in the questions box, that would be really helpful for us. And with that, I'm going to introduce Nick Dugan. He is our first presenter today. Nick is an environmental engineer with our Office of Research and Development in Cincinnati, Ohio. And he will be talking about treatment options. And Nick, I'm going to go ahead and turn that over to you.
Thanks, Michelle.
Hey, Nick, you're going to have to mute your speaker, just so you know.
Hold on.
Okay.
Am I muted?
Yep, sounds good.
Great. Let's see here.
Looks good too. Your screen looks good.
Great. So let's dive into. So I'm going to talk about. Conventional surface water treatment, and we are going to, over the course of the presentation, talk about what happens during. We're going to talk about what happens during during coagulation, flocculation, and sedimentation. We'll talk about what happens during filtration. We'll talk about what happens during chlorine addition. And we're also going to talk about a little bit about powdered activated carbon. We're going to talk about a little bit about potassium permanganate. And we're going to dive into some new data on what happens when suspensions of cyanobacterial cells in the source water have been exposed to copper sulfate algicide and then subsequently treated with potassium permanganate. And then if we have time, we're also going to talk about some different ways of tracking the condition of cyanobacterial toxins and condition of cyanobacterial cells through the treatment facility. So let's dive into some definitions. Chlorophyll is a pigment molecule that's common to both algae and cyanobacteria, and that molecule plays a role in photosynthesis. Microcystins is it's one of the major groups of cyanotoxins that is produced by cyanobacteria, and it's one of the more commonly analyzed for and detected toxins, and one of the target organs is the liver. When we talk about ELISA, we're talking about the enzyme-linked immunosorbent assay, and it's an assay that depends on specific antibodies to detect various types of cyanobacterial toxins. When we talk about LC tandem mass spec, or LC-MS-MS, we're talking about liquid chromatography followed by tandem mass spectrometry, and it's another way of analyzing for cyanobacterial toxins in solution. During this presentation, when we talk about membrane-compromised cells, we're talking about cyanobacterial cells whose membranes can be permeated with a stain called Cytox green. And typically, it's accepted that this is a stain that will permeate the membranes of, permeate cell membranes that have been compromised, have been structurally altered in some way. Here is a hypothetical water sample, and here is a cyanobacterial cell. And when we talk about intracellular toxin, we're talking about toxins during this presentation. This will mean toxins that are actually contained inside the cell membrane. When we talk about extracellular toxins, we're talking about toxins that are in solution, in aqueous solution in the water in which the cell is suspended. When we talk about total toxins, for this presentation, we're talking about the sum of extracellular plus intracellular toxins. So let's move into the core of the conventional surface water treatment process, so coagulation through filtration. And in this slide, we're looking at cell propagation of cyanobacterial cells through a full-scale treatment facility in Lake Erie. On the x-axis, we have the different treatment stages. And on the y-axis, we have the concentration of chlorophyll A in micrograms per liter. So that's on both of the y-axes. And what we're showing here are three different profiles, through plant profiles of cyanobacterial cells collected on three different sampling visits. And the chlorophyll, the way we got at the chlorophyll concentration is we took the water sample and we filtered it through a glass fiber filter. And so then we took what was retained on the filter and then we extracted the chlorophyll from it. So the chlorophyll number is essentially a proxy for the concentration of cyanobacterial cells in suspension. And the highlight, the take home, is that from this slide is that most of the cell removal work is accomplished prior to filtration. So if you look at the raw water chlorophyll concentration, and then you look at the filter influent chlorophyll concentration, you can see that we achieved greater than 2 orders of magnitude removal of cyanobacterial cells before the water ever got to the filters. And so when you think about your treatment process, that's kind of like what you want to shoot for. You want to have, your filters. You don't want to be depending on your filters, except ideally as kind of a cleanup step in the treatment process. You really want to let your clarifiers do most of the work. Here, this is a summary slide of a sampling campaign that we did along Lake Erie. We looked at 7 full-scale treatment facilities along Lake Erie. And on this slide, on the graph, we have facilities 1 through 7 that were arranged on a transect that was about 150 miles long from west to east. And on the y-axis, we have the concentration of chlorophyll A. And then each of these series summarizes the physical removal of cyanobacterial cells through the treatment facility, through an individual treatment facility over the course of the sampling campaign. So each data point represents the average of 5 to 7 samples collected between May and October. And each circle represents an average of the influent concentrations. Each triangle represents an average of the settled water concentrations. And each square represents an average of the finished water concentrations over the course of this particular bloom season. And the take home from this graph is that from influent water to settled water, we see consistently on the order of a log to more than a log of removal from influent water, so through the clarifiers. So once again, the bulk of the cell removal is being done in the clarifiers, and then in most cases, all right, the filters are basically acting, kind of as a cleanup stage. Here's some work that was done by folks in Australia, and just as an aside, throughout this presentation, it's going to be mostly work that was generated by the EPA, but we do include some work from the peer-reviewed literature to kind of round out the presentation. In this case, this is a combination of full-scale through jar test data that was generated in Australia. And let's see, we have the percent removal of cyanobacterial cells on the x-axis, and up here, first we have the full-scale, through a full-scale facility with a high dose of polyaluminum chloride coagulant, and they observed anywhere from 55% to almost 99% removal of cyanobacterial cells through coagulation and sedimentation. Here we've got some, in the middle, we've got some pilot scale data, 70 milligrams per liter of alum, and they observed somewhere between 70 and about 85% removal. And then in a jar test with 65 milligrams per liter of alum, it was pretty predictive of the pilot scale results, showing 70 to just under 85% removal of cyanobacterial cells. Here's some more data from Australia. We're looking at toxin removals through pilot scale, coagulation, sedimentation, and filtration. And we have, we're looking at the results of two trials, trial 1 and trial 2. And here the variable is the concentration of microcystin LR in micrograms per liter. And we're looking at both the total toxin and the extracellular toxin in the influent and the effluent. And so we can see that the influent concentrations for total toxin ranges from 60 to 120, almost 120 micrograms per liter, while the extracellular toxin concentrations range from 2 to 3 micrograms per liter. And through treatment, we can see the total toxin is decreased from 120 to 3 micrograms per liter in trial 1 and 60 to 2 micrograms per liter in trial 2, while we see very little change in the extracellular toxin concentration. And so this kind of summarizes where you want to be with conventional treatment, okay? So you want to remove your toxin burden. You want to remove you want to achieve as much physical removal of the cells as you can, while keeping the toxin inside the cells and not letting it get through, not letting it leak out of the cells and into solution. Now, let's move on to powdered activated carbon. And powdered activated carbon, you take advantage of the porousness of the activated carbon. And so your contaminants basically partition from the liquid phase into the solid phase by adsorbing to the surface of the carbon as you go, taking advantage of the huge surface area that is provided by the high porosity of the activated carbon. And the effectiveness of the powdered activated carbon is pretty site-specific, okay? So it depends on the kind of carbon, wood or coconut or coal-based carbon. It depends on the kind of toxin you're trying to adsorb. It depends on the carbon dose and the carbon contact time. And it depends on how much natural organic matter you've got riding along in solution and potentially competing for adsorption sites. And jar testing is really your best bet for assessing the kind of assessing the kind of powdered activated carbon you want to use and the dose that's going to be required. And here's an example of a jar test. This is one that was done in our laboratories, and we're looking at the impact of powdered activated carbon addition. This is a wood-based carbon, it's a coal-based carbon, sorry. So we've got contact time in hours on the x-axis, and we've got extracellular microcystin LR concentration on the y-axis. We dosed the water, it's a natural water. We dosed it with about 65 micrograms per liter of toxin, and then we added 10, 20, 30, or 40 milligrams per liter of PAC. And the first take home is that as you increase the dose of carbon, you do see increasing removals of extracellular microcystin at any given time point. And then once you get to the 20 to 40 milligrams per liter carbon doses, you see that for a given carbon dose, as the contact time increases, so does the amount of removal of toxin from solution. Here we're looking at, in this slide, we're looking at what happens when carbon is added after toxin has been released from cyanobacterial cells. So these are experiments where we added potassium permanganate to suspensions of cyanobacterial cells, and we observed at 2 different pHs, pH 7 and pH 9. And we observed toxin release over the first 30 minutes of exposure time. And then we added powdered activated carbon. And then we looked at what happened when they're with carbon present and without carbon present. And we can see that at pH 7, adding the carbon over the last 60 minutes resulted in significant decreases in toxin concentration. And we observed the same thing at pH 9. Let's move on to oxidation. And in this chart, this is a chart that we would like to thank the Ohio EPA and the Ohio section of the American Water Works Association for putting this data together. And this shows the impact of common drinking water treatment oxidants, so chlorine, chloramine, chlorine dioxide, potassium permanganate, ozone, and UV, and how effective they are against the 4 major classes of cyanobacterial toxins, so antitoxin A, cylindrospermopsin, microcystins, and saxitoxins. And we'll focus in on the microcystins. And so the microcystins, it's effective against chlorine. It's not, chlorine is effective against microcystin. Chloramine is not at normal doses used in drinking water treatment. Neither is chlorine dioxide. Potassium permanganate is effective. Ozone's effective, and UV is effective, but it's effective at doses that are much higher than what's used for cryptosporidium control. And let's focus in the next slide, we're going to focus in on the chlorine and talk about why show how it's dependent on a pH and temperature for its effectiveness. So in this slide, we're looking at the impact of pH and temperature on the effectiveness of chlorine and how effective it is with respect to removing microcystin LR. So we have pH from 6 to 8 on the x-axis, and then we have the chlorine CT that's necessary to reduce microcystin concentrations from 10 micrograms per liter to 1 microgram per liter, so to achieve 90% removal of microcystin LR at various pH and temperature combinations. And we looked at 4 different temperatures, 10, 15, 20, and 25 degrees C. And let's look at, for example, 25 degrees C, and we can see that as the pH increases, so does the CT that's necessary to achieve 90% removal. And then at any given pH, you can see that as the temperature, let's take pH 8, for example, and we can see that as temperature decreases, so does the CT that's necessary to achieve 90% removal. And just for perspective, we also included the CT that's necessary, that's required under the surface water treatment rule for 3 log inactivation of Giardia at a 1 milligram per liter chlorine dose at 25 degrees Celsius. Now, let's look at what happens when our source water is treated with copper sulfate algicide, and then when that algicide exposure is followed by exposure to potassium permanganate. And the reason we looked at that is that's a scenario that's that's a scenario that could conceivably occur early in the treatment process. So you have a reservoir, you have an algicide that's added to a suspension of cells in the reservoir, and then as the water moves into the treatment plant, it is potentially exposed to potassium permanganate early on because potassium permanganate is typically one of the first oxidants, one of the first treatment chemicals that's often added in the treatment process. And so to look at this, to start looking at what happens, we designed a series of experiments to look at the impact of algicide exposure. So we looked at 3 different pHs, 6, 7, and 8, we looked at 3 different copper sulfate doses ranging from 0.1, 0.5 to 1.0 milligrams per liter as copper. And then we looked at, so we had 3 pHs and we had 3 copper sulfate doses. So we looked at 9 combinations of pH and copper. And at each of those combinations, we evaluated the impact of 24 hours of exposure to copper sulfate, followed by 90 minutes of exposure to potassium permanganate at an initial dose of 1 milligram per liter. And what we're going to do here is we're going to highlight what we saw at pH 7 and a 1 milligram per liter, so 1 milligram per liter copper sulfate dose at pH 7. And in this first slide, we're looking at extractive chlorophyll A. So that's chlorophyll where we've taken the sample, we've taken a water sample, we've run it through a glass fiber filter, then we extract the chlorophyll that's in the cells in the cells that were retained on the filter. And we're looking at 3 sets of data in this graph. We're looking at a control where no copper sulfate or oxidant was added. We're looking at where then some data where oxidant only was added, and we're looking at a combination of copper sulfate exposure followed by oxidant exposure. And so this is 0 hours. It's immediately prior to copper sulfate addition, and here is our initial chlorophyll concentration. Then at 24 hours, this is after 24 hours of copper sulfate exposure and immediately prior to the addition of 1 milligram per liter of potassium permanganate. And then finally, we have what happens after 24 hours of copper sulfate exposure, followed by 90 minutes of permanganate exposure. And we can see that in the 2 jars where no copper sulfate was added, we can see the chlorophyll concentrations are significantly higher than in the jar where copper sulfate was in the jar where cells were exposed to copper sulfate. And then we added potassium permanganate, and we can see that after adding potassium permanganate, we can see that in the oxidant-only jar, we had a decrease in extractive chlorophyll of about 17 micrograms per liter, while in the jar that had received copper sulfate exposure and permanganate exposure, we see a decrease of about 8 micrograms per liter. So the impact of copper sulfate is profound, but adding the permanganate, we see an impact of permanganate, but it looks like prior exposure to copper sulfate, in this case, does not result in a real change in how the cells then react to permanganate exposure. Here we're looking at the impact of what happened with in vivo chlorophyll A. So in vivo chlorophyll A, this is just a straight fluorescence assay. We take a water sample and we used a Turner benchtop fluorometer, and we looked at the fluorescence of chlorophyll A using the excitation and emission frequencies that are specific for chlorophyll A. And we can see that in the 2 jars. So here we are. Prior to copper sulfate exposure. And here are the 2 jars that were not exposed to copper sulfate. And then after 24 hours, down here is the jar that was exposed to copper sulfate. So we can see that adding copper sulfate had a significant impact on in vivo chlorophyll fluorescence, where that's measured as relative fluorescence units. So adding copper sulfate had a pretty significant impact on in vivo fluorescence. Then we added potassium permanganate to the oxidant-only jar and to the copper plus oxidant jar. And we can see that after 90 minutes of permanganate exposure, we don't see a real big change in the in vivo chlorophyll fluorescence. So once again, adding copper sulfate had a big impact, but then the subsequent addition of potassium permanganate, the impact, there wasn't really a discernible difference with respect to whether the cell was exposed, whether the cells were exposed to copper sulfate or not prior to permanganate addition. So let's look at what happens to total microcystin. Prior to any type of chemical exposure, we have total microcystin concentrations ranging from 25 to about 32 micrograms per liter. Then after 24 hours, in the jars that were not exposed to copper sulfate, there's no real difference between them. While adding copper sulfate did result in a significant decrease in the concentration of total microsystems. So from roughly 10 microgram per liter difference. And then after adding potassium permanganate, we can see that total toxin concentrations actually increased. Total toxin concentrations increased. Remember, this is the sum of extra plus intracellular toxins, and they increased in both of the jars that received oxidants, about 2 micrograms per liter for the oxidant-only jar, roughly 5 micrograms per liter for the copper plus oxidant jar. So once again, the takeaway here is that copper sulfate addition had a significant impact, and we had a less discernible impact. We had a smaller difference when after we added the oxidant. So prior copper exposure didn't really seem to make a huge difference once we added the oxidant. Here we're looking at extracellular microcystin. So in all cases, extracellular microcystin was right at the reporting level just before copper addition. And once we added copper sulfate after 24 hours of copper sulfate exposure, extracellular toxin concentrations were higher in the jars that had received copper sulfate. Extracellular toxin concentrations were higher. Then once we added potassium permanganate, we saw 0.8 micrograms per liter decline in the oxidant plus copper jar compared to a 1.3 microgram per liter decline in the oxidant-only jar. So once again, we had a large, so the impact of copper by itself was large. The impact of copper on what happened after oxidant exposure was smaller. Here we're looking at the ratio. Here finally we're looking at the ratio of extracellular to total microcystins. Prior to copper sulfate exposure, almost all the microcystin is inside the cyanobacterial cells. Then after 24 hours, the jars with no copper sulfate concentration, roughly 10 to 15% of the toxin was in the extracellular form, while the jars where the cyanobacterial cells were exposed to copper sulfate, roughly half the toxin had leaked from the cells in the solution, and then we see that after permanganate exposure, we see a decrease in extracellular toxin concentration as the permanganate reacts with the microcystin. Moving on, and we'll spend the last few minutes of my presentation looking at some different ways of assessing toxin concentrations and cell integrity during treatment. And what this data is generated. We ran a series of experiments where we looked at the impact of potassium permanganate application at 1, 2.5, and 5 milligrams per liter permanganate dose at pH 7 and 9, and we looked at turbidity and at waters with turbidities of 0.1, 5, and 20 NTU, permanganate times of 0 to 90 minutes. And we looked at extracellular toxins, we looked at total toxins, and we measured toxins by both ELISA and by LC mass spec. And the slides that follow, control is going to indicate cells, cyanobacterial cells that were not exposed at all to potassium permanganate. And oxidant exposed indicates cells that were exposed to permanganate at CT values ranging from 0 to 400 milligrams per minute, 0 to 400 milligrams per minute per liter. And the oxidant residual is always quenched at the time of sample collection. So here, this is the concentration by ELISA, micrograms per liter on the x-axis, and concentration by LC mass spec on the y-axis. And here we see the correlation that we observe between the two methods for control samples, no oxidant addition at 0.1 NTU turbidity. Then when we add in when we pool in the 5 NTU turbidity control jars, then we also pool in the 20 NTU turbidity. Then we start pulling in oxygen exposure data. Then we start pulling in oxygen exposure at 5 NTU. And finally, when we pull all the data together, control plus oxygen jars at ranges at CT constant at CTs ranging from 0 to 400 milligram per minute per liter at 3 different turbidities. And when you pull all that data together, we see a correlation coefficient of 0.85. And when you dive into the data, what you should look at here, you can see that whether we look at oxygen exposure only, control jars only, or oxygen control data pooled together, and whether or not it's broken down by pH of 7 or 9, or turbidities of 0.1, 5, or 20 NTU, what's important to note here is that you have high correlation coefficients. So not only are they high, but they also are statistically significant at the 99% confidence level. And so the take home here, the way to think about this physically is that if you're noticing a change in toxin concentration measured by ELISA, if you're noticing, for example, an increase in toxin concentration as measured by ELISA, then there's a high probability that you're also going to see an increase in toxin concentration measured by LC mass spec. Now here we're looking at the same data, we're looking at ELISA versus LC mass spec, but for total toxins. And we start with control jar 0.1 NTU, and we work through the progression of increasing turbidities with the control jar 0.1 to 20 NTU. Then we start adding in the permanganate exposure data at the 3 different turbidities. And so we're seeing lower correlation coefficients, and that has to do with the fact that we just observed more scatter in our total toxin data. There is more variability in our total toxin data as opposed to our extracellular toxin data. But when you dive into the correlation coefficients, you can see that they're all, for the most part, still greater than 0.5, and that that they're statistically significant at the 99% confidence level. And so once again, what that tells you is if you're measuring total toxin concentrations by ELISA and you're seeing an increase, then there's a high probability that your total toxin concentrations measured by LC mass spec are going to move in the same direction as what you saw the ELISA concentrations move, the ELISA assays move in. Finally, we're going to look very quickly at assessing cell integrity and toxin concentrations during treatment. And we look at the concentration of membrane-compromised cells versus the ratio of extracellular to total toxins, in this case, microcystins. And we looked at some membrane-compromised cells are the cells that the membrane passes the cytokine stain. And this was based on simple cell counting and green fluorescence and cell fluorescence. We're looking at cyanobacteria concentrations ranging from 0.5 to 1 times 10 to the 6 cells per milliliter. And we're looking through the progression, control jars, no oxygen exposure. And then once we start exposing cells to an oxidant, right, we're seeing significant increases in the concentration of in the extracellular to total microcystin ratio. And so as the concentration of membrane-compromised cells increases, so does the corresponding ratio of extracellular to total microcystins. When you dive into the correlation data, especially once you look at the cells that were exposed to potassium permanganate, you see reasonably high correlation coefficients, and they're all statistically significant, which once again tells you that if you're tracking this kind of stuff and you're seeing an increase in the concentration of membrane-compromised cells, there's a good probability that you're also going to see an increase in the extracellular and the ratio of extracellular to total microcystins, which means you're starting to see toxins being released in the solution, and it means that you need to take a closer look at what's happening. So moving on to our conclusions, really quickly, when optimized, conventional treatment processes are highly effective at removing cyanobacterial cells. Powdered activated carbon will effectively absorb microcystins. Common drinking water treatment oxidants are going to degrade toxins, but the effectiveness can depend on matching your oxidant with the right water quality. Chlorine is effective at degrading microcystins, but it's temperature and pH dependent. Ozone effectively degrades microcystins. And chlorine dioxide, chloramines, and UV at the dose levels commonly employed in drinking water treatment are not effective against microcystins. Permanganate does degrade microcystins, but vigilance is recommended because permanganate is employed early in the treatment process where there is a potential for toxins to be released from cyanobacterial cells. Copper sulfate exposure has a significant impact on cyanobacterial cells. However, the impact of permanganate on cells exposed to copper sulfate is less clear. ELISA and LC mass spec measurements of extracellular and total microcystins are correlated across a broad range of pH turbidity and permanganate CTs, and the concentration of membrane-compromised cells is correlated with the extracellular to total microcystin fraction measured by both ELISA and LC tandem mass spec. And with that, I'd like to hand the presentation over to Joel Allen. Thank you very much for your time and attention.
All right, great. Thanks, Nick. Good job. Hopefully everyone can hear me.
Yes, we can hear you, Joel.
Oh, great. Thank you. I was waiting for somebody to give me the go-ahead. All right. So I'm going to talk about harmful algal blooms from a little bit different perspective than what Nick presented. My background is as an aquatic toxicologist, so my area of interest is more on the source water side. Typically, when we think about harmful algal blooms, there are a couple of major drivers which are very difficult to manage, those being increased temperatures and excessive nutrients in our source water systems. What I'm interested in looking at is how do we examine the underlying ecological drivers for these harmful algal blooms, and using essentially off-the-shelf type technologies to essentially give a heads up, an early warning as to when we might expect an increased likelihood of a harmful algal bloom event. See if I can move to the next slide. There we go. So essentially, we're wanting to predict cyanotoxin, cyanohab/toxin events. So I make that distinction because you can observe cyanohab events where toxins are not being produced. And of course, we are looking at these events through a sort of regulatory lens, in that there are advisories and criteria out there that set concentration limits for consumption or recreational exposure. So really the key question is how can we determine when we are going to have not just a cyanohab event, but a cyanotoxin event. There are quite a few different tools available for gathering information regarding cyanohab events, ranging from sort of traditional wet chemistry approaches through high frequency data collection and molecular techniques. And we have the good fortune here in Cincinnati of having folks that are expert in each of these. So we've been able to put together a research team that's been able to leverage different areas of expertise, and we're beginning to put together an interesting story, at least what's happening in our neck of the woods. The goal being to provide a sort of a near-term local predictive capability based on what's going on in your source water. There are varying scales of sort of spatial and temporal monitoring techniques. This is a very local approach as opposed to looking at satellite imagery and trying to determine HAB events that occur across larger areas and perhaps with a less frequent sampling interval. Ideally, we would like to be able to get to the point where we can be looking a week to 2 weeks into the future to provide actionable information for drinking water utilities and other stakeholders. So for our study, this is a list of the tools and techniques that we are using. We have high-frequency physicochemical monitors, essentially data sondes. Give you really good background on sort of what's going on in your system, especially with temperature being a very important parameter. And along, with these sondes, you can get at your algal community as well, and I'll touch on that in just a second. We do wet chemistry, so we try to track what's going on with our nutrients within the system. The phototroph dynamics, in vivo fluorescence has really come a long way. We can not only determine whether there is green stuff out there, but we can also determine whether that green stuff is a green algae or a cyanobacteria. So being able to make that differentiation is quite helpful. The molecular techniques are also very interesting, especially when we can look at the specific activity of the genes responsible for cyanotoxins. We have folks that are looking at ribosomal RNA for sort of cataloging what's in our system, and also levels of activity with the rRNA. And then looking at qPCR and RT-qPCR techniques to sort of quantitatively get at when, in our case, the microcystin producing genes are active. And of course, finally, we do the analytical side. We look at ELISA as well as LC-MS. And as Nick mentioned, they can at times be a bit of apples and oranges, particularly with the microcystins and the number of congeners that can be observed within a system. Just briefly, a little bit about the system that we're studying. We're located in Cincinnati, Ohio. There's an Army Corps reservoir, managed reservoir in Claremont County, which is just down the road. It drains 340 square mile watershed. Summer pool elevation 733 feet. What that works out to be is anywhere from 10 10 to 20 feet in sort of the mouth of the river coming in, up in this area, all the way to over 100 feet where the dam and spillway are located. 2,000 acres, so not that big of a reservoir, but somewhat typical for what we find, at least in the Midwest. We do have the advantage of a historical data set. Data have been collected at relatively frequent intervals, 3-week intervals, since 2012. We have multiple sampling sites. We have 2 buoys that are located, one near where the river, East Fork Little Miami River enters the reservoir, and another one in essentially the western basin. And we're also monitoring near the drinking water intake, and then a beach area. The reservoir is surrounded by a state park, so there's really no residential type of improvements around the reservoir itself. So one of the questions that really started piquing my interest was, if we look at this plot, this is measurements of phycocyanin using a data sonde. These were collected in 2014. Over the time units are normalized, but this is essentially over the first 3 weeks of June in 2014. And the top, oh, my zoom went away. In the top panel, that is the raw data. So as you can see, over the course of this time period, we had phycocyanin that was relatively low at the beginning. Of course, it increased towards the end of the period. And of course, if you're out at the lake looking at it, you can observe this lake going from brown to green, and it's quite striking. But what was really interesting to me was the fact that when you decompose this signal, you can look at the periodicity. This is every 24 hours. So we've got a cyclical occurrence here where we're seeing changes in our cyanobacteria density at the surface where this measurement was being taken place. And I'll come back to that a little bit later. Another thing that is really interesting when you look at monitoring sort of through the lens of how often are we monitoring, and whether or not we are sort of tracking the biological activity of the system. Traditionally, you might go out and sample once a week, once every 2 weeks, once a month, and a lot of things can be going on. These data are from 2015, and you can see that over the course of that period between June and July, we've got days where there are significant hits when you might not expect it. So we've got microcystin concentration on the X, and unfortunately at this time we were not making dilutions of our sample. So anything that's up here in the purple, those are concentrations that essentially blew our standard curve. So all we can say is really that they're higher than 5 micrograms per liter. And we have one about the 6th of June, which we were really scratching our heads when we saw this, what we thought at the time might be an outlier, when in fact it was corroborated with LCMS data. And when we look at the taxonomic data, we can see kind of what was driving that. So really, the take home from this slide is that time matters, particularly when you're thinking about how and when you're going to do some sampling for monitoring. And just real quickly, we'll go over some of the different various data that we do collect. For the high frequency data, you know, there's nothing that's terribly earth-shattering with this. The data pretty much follow what you might expect. If you look at this as a series of panels, two series of panels, the top series is 2015, the bottom series is 2016, and there are differences from one year to the next. In 2015, we had relatively high phycocyanin, which is the BGA PC raw, for a much longer duration in 2015 than we saw in 2016. Our temperatures were fairly consistent, and so we know with our system that once we hit beginning of June, we're coming right up on it. We can expect to see a bloom once we get above about 23 degrees out in the lake. Now I want to touch on the taxonomy that we observed. Lake Harsha is a relatively diverse reservoir. In speaking with some of the state of Ohio folks, they really don't see this kind of diverse taxonomic diversity in other reservoirs that are being monitored. We typically see an Anabaena bloom in the beginning of the summer, and that then sort of transitions through Pseudoanabaena and Merismapedia. And the red bars are the microcystis, and this is relative abundance of cells. And the microcystis does not necessarily, it's not around consistently throughout the season. And we do observe that we see our highest concentrations of microcystin when microcystis is present. And microcystins are actually produced by many other taxa beyond microcystis. So you might expect to see higher concentrations throughout, but we're really seeing that microcystis is our most important player. Just some of the ELISA data that we've collected over the years. Again, where we have these purple dots, those are the samples that blew our curve, and so we can only say that it was greater than 5 micrograms per liter at that point in time. And again, 2015, 2016, we saw different things happening. We had high concentrations in 2015 across the sites. The middle site, EFLD, that is actually taken at depth of about 25 feet. It's where the local drinking water utility is drawing their water from. So it's interesting that we have high concentrations at our surface sites. Of course, much lower concentrations at depths below essentially the thermocline. But throughout the year of 2015, we had relatively high concentrations. And then 2016, we had more of this peak associated with the major bloom event that occurred early in the year. And just a sampling of some of the other types of data that we're collecting, ribosomal DNA and RNA sequencing, looking at cataloging what is there and what the relative activity is. We're essentially dominated by the nostocs, and that's obvious in our ID taxonomy as well. And of course, being able to sort of build a weight of evidence that indicates sort of what's going on in the system is important. qPCR and reverse transcript, easy for me to say, reverse transcriptase qPCR. So this is the technique that's really looking at the specific genes that are responsible for, in this case, microcystins. In this top plot, we have the qPCR. So really, what we're seeing here is as the genome count on the X increases, we're also seeing an increase in the microcystins measured with ELISA on the y-axis. And we do see a very good relationship between these two. And this is telling us, so that the gene count is telling us how many replicates of the MCYE gene is present in our samples. Of course, the correlation is that as your population density is increasing, that genome count is also increasing. The power of this approach is that it based on these results, that you have taxa that are capable of producing microcystins in your system. And if we just quickly at the looking at the bottom plot here, again, we've got gene count on the X and the ELISA concentration on the Y. And this is the qPCR or RT qPCR. So we're looking at activity, and the activity of those of that portion of the microcystin gene. Again, we've got very good correlation between concentration of the toxin and the genome count. And I won't really get into it now, but we're looking at different markers which look at different sections of the microcystin gene complex, as well as those that are specific to different taxa. So the first slides that I showed were a portion that's conserved generally across all microcystin producers, and then the two panels on the right. This is one that is specific. This is a marker specific for microcystin, or microcystis, sorry. And so, again, very good correlation between what we're seeing in the counts and concentration.
Hey, Joel?
Yeah.
Sorry to interrupt you. This is Michelle. I'm going to have to jump in here for a minute because we are at almost 3 p.m.
Okay.
Eastern. So we'll let you finish your presentation, but I just
Sure, I can wrap up real quick.
Okay. All right. Just those on the line. Once Joel wraps up, we will start the Q&A session shortly after. Okay, go ahead, Joel.
Sure. Yep. The upshot of these data are that we really feel like the molecular indicators are quite powerful to describe your system, and really in getting a basic understanding of what is going on in your source. And year over year, we're seeing very similar results. So it may be that from a management perspective, you want to establish what is going on in your system, what players are coming to play, and have that as a basis for monitoring and management activities. Let's see. I'm going to real quickly. I want to hit this. Start over here. So again, this I'm going to go back to when we observed the 2015, the very early June hit of the high concentrations of microsystems. And if we look here on the biovolume density, this is a little bit different measure than what I presented earlier. Microcystis is red. It's hard to see microcystis in there. So the take home being that you don't have to have a huge microcystis bloom to be able to observe large concentrations of microcystins being produced by the microcystis community. Looking at the sort of the spatial distribution of our communities, when you're out there and you look at these blooms from day to day, they start to have a little bit of a personality. This is a photo that I took at 10:50 at night, and you can kind of see these particulates in the water. Well, those are moving, and they're moving up or they're moving down. So we know that some cyanobacteria are able to move, change their elevation within the water column, and it's really quite striking when you're out there and you can see it occur. So the question being, what's the impact on our monitoring regimes? So do we need to go out at a certain time of day? Do we need to be cognizant that these concentrations may change as these organisms are moving up and down? So real quickly, this is we were out and did a 24-hour study where we took samples every other hour, and we were looking at do our concentrations change over time? And when we look at our surface, so if we look across the top, each panel or each box is a profile at that particular hour of the day. So at 4 p.m. we started, and we had concentrations that were sort of in the middle of our X range. And then as we moved into the evening and early morning hours, we shifted to higher concentrations at the surface. And this is during a period when Anabaena or Dolichospermum was dominant. So we did observe changes in concentration at the surface throughout that 24-hour period. We did another one later in June, and we actually saw higher concentrations at lower depths than we saw at the surface. So we were seeing higher concentrations between 2 and 3 meters below the surface, and I'm not sure that typically sampling is occurring at those depths. I think typically we maybe stick a bucket in or a bottle in as far as our arms will go, but most often I think we're probably just dipping from the surface, and that might not be telling the whole story. And a third event, again, we had maximal concentrations at much lower depths than just at the surface. So finally, in summary, we have a very nice study area that gives us harmful algal blooms every year, so we can keep going back and taking a look at what's going on. A great team of researchers that bring a lot of different tools to the table, and we've been able to sort of conclude that that sample timing and location do matter, and we're moving on towards being able to make some predictions about when we're more likely to observe HAB events, and particularly toxin-producing HAB events.